Welcome everyone, my name's Chris Edgar and I'm the Senior VP Clinical Science with Cogstate, and today I'm going to be talking about the use of cognitive assessments as efficacy outcome measures. Unlike other measurement concepts in drug development, cognition and cognitive test performance is measurable in both healthy and patient populations. We know that cognition may be impaired as a result of a variety of disease states. It may also be impaired by drug treatments, and also impaired by factors like fatigue or sleep deprivation. And because of these factors, cognitive tests can be relevant across all phases of clinical drug development and also be applied to answer multiple different kinds of questions that we might pose. In respect of efficacy then, the context of use for cognitive tests include both early signal detection and also later evidence of efficacy. When we think about early clinical development, go, no-go decision-making is really key there. Here, cognitive tests can provide sensitive tools that enable us to detect efficacy signals in relatively small studies that may also be of fairly short duration or conducted in healthy or less clinically impaired populations. Properties of optimal tests then include things such as brevity, reliability and stability with repeated assessment. And also these factors help us to further facilitate the detection of efficacy signals early on. These data then can be incorporated into decision-making frameworks and used to guide decision-making regarding progress to later phases of development. In order to increase our confidence in decision-making, it may be necessary to look for both convergent lines of evidence, but also relevant treatment effect sizes. As an example then, here we can see a study where consistency of effect was focused upon and a signal for a cognition assessment outcome or parameter was used to provide support for a go decision in further development. Typically, we see that batteries of cognitive tests are employed at early phase where they have multiple parameters. And in these kinds of cases, go criteria might be developed that deliberately include a need for an effect to be identified on more than one outcome measure. But equally, we might see that there are inclusions of other modalities of testing, for example, things like quantitative EEG. And here, a signal was seen on both a working memory test and also the QEEG. And by this convergence of two different lines of evidence, greater confidence was seen in terms of being able to make a go decision. And then there are also other emergent lines of evidence that are brought to bear around evidence for target occupancy and effects on exploratory biomarkers, such that there was increased confidence then to move on to phase 2B studies with the compound. Additionally to convergent lines of evidence, effect size can be an important metric to incorporate into go, no-go decision-making. We know that drugs are not approved based on statistical significance alone, and we need successful treatments to be able to demonstrate sufficiently large effects such that they confer clinical benefit to patients. Effect size at the level of between groups comparisons has been a very common metric for clinical trials and is often what we use in meta-analyses of trial data across different studies. And several different approaches to interpretation of effect size have been proposed. We know that Cohen initially proposed effect size interpretation from small through medium to large effects. Although, of course, that was immediately qualified in highlighting that effect size as a relative metric, and under certain circumstances, smaller effect sizes may still be relevant. When we look at reviews of clinically important differences, they tend to converge around effect sizes of between 0.3 and 0.5 as being clinically meaningful. And we also know that from approved drugs, you tend to see a fairly common range of effect sizes, such that in psychiatry drugs, for example, approved drugs fall into the range typically of 0.3 to 1 in terms of effect size. And so we have lots of converging lines of evidence that point us towards an effect size threshold of around 0.3 as perhaps being a lower bound for clinically important differences. And this might provide another useful guide for go, no-go decision making. Additionally, in early development, we may apply models of cognitive impairment that can be useful in elucidating effects of certain kinds of treatment when they're intended to be cognition enhancers. A common example here is the Scopolamine challenge model, where scopolamine is used to induce a cognitive impairment, either in animals or humans, through the blockade of central muscarinic receptors. We know that muscarinic receptor blockade, either via atropine or scopolamine, is well known to produce memory impairment, but also other forms of cognitive impairment. And using scopolamine, we have a safe and reproducible model of cognitive deficits, such as those that are seen in Alzheimer's disease. We can then explore the potential efficacy of drugs for the treatment of AD and other forms of cognitive impairment, and look at the ability of putative cognition enhancers to reverse the effects of scopolamine. We can see here an example of a study which has used multiple crossovers in order to explore the enhancement of histaminergic neurotransmission, as well as cholinergic neurotransmission, and the combination of the two in attempting to reverse cognitive impairment that's been induced via scopolamine. And we can see here a reliable and robust reversal of cognitive impairment on a hidden pathway maze learning test designed to assess executive function. And these kinds of data too can give us increased confidence that drugs are having the expected pharmacodynamic effect and provide a strong early signal of potential efficacy. When we start to think about later clinical development, cognition may also be a target for specific studies of efficacy. And here, cognitive tests can be incorporated in a number of different ways in terms of the hierarchy of outcome measures and endpoints included in a trial. They may be exploratory or secondary efficacy outcomes aimed at more fully characterizing clinical benefit, or they may even be primary or co-primary endpoints, where cognition is an important target for improvement of particular diseases. It might also be possible to develop cognitive tests as potential surrogate or intermediate endpoints, where the measures might be used to predict something like disease progression or later functional outcome. When we start to think about evidence of clinical benefit, we're really talking about how people feel, function, or survive. And in this context of assessment, the outcome measures that we use to define such endpoints are termed clinical outcome assessments. As measures of task performance, cognitive tests here are a subset of clinical outcome assessment that we term performance-based outcome assessments, or PerfOs. And these PerfO assessments broadly include measures of things like physical function, so for example, mobility, or a walking test. They could also include things like sensory function, like visual acuity, and of course, as we said, cognitive function, where we may assess tests of things like working memory, episodic memory, and attention. When we use cognitive tests as clinical outcome assessments to confirm evidence of clinical benefit, we need to then be careful to confirm all of the relevant components of validity, reliability, and ability to detect change, as we would with other clinical outcome assessment types like PROs, clinician-reported outcomes, and also observer-reported outcomes. So thinking then specifically about supportive efficacy, here we might use performance-based outcome assessments to understand particular components of additional efficacy that a drug treatment may be providing. It's often useful to think about performance-based outcome assessments where self-report may be considered less reliable, or observer report might be impacted by bias. And here we can see examples where patient-reported outcomes or observer-reported outcomes may be considered insufficient evidence of efficacy. And that might be in populations where the population themselves have some degree of cognitive impairment, such as Alzheimer's disease patients, and two, where study partners, caregivers, might also have some degree of impairment in terms of ability to report, or some degree of bias. But also we see other populations like pediatric populations where it's very difficult for them to self-report health status, or where we may be concerned about looking to support reports that come from parents with some objective evidence. An example of this in terms of guidance is the recent FDA draft guidance in rare disease, where FDA consider performance-based outcome assessments of things like cognition and ambulation as complementing reports from caregivers and patients regarding relevant aspects of function. A particular example of current use of cognition as a secondary efficacy outcome measure is in multiple sclerosis trials. Here we see that cognitive and motor performance measures are commonly being employed where the purpose is to determine another aspect of efficacy of the treatment. An increasing focus of studies in multiple sclerosis has been the single-digit modalities test, recognized as being a particularly sensitive measure of information processing speed in the population. And this is really a basic element of cognitive function likely to influence downstream processes and important to functional outcomes in the patient population. We can see here an outcome from a large clinical trial where the single-digit modalities test has been included to measure processing speed, a frequently affected cognitive domain in the patient population, and where, as an exploratory outcome measure, a positive outcome has been seen in terms of ability to improve performance on the test. And this then is potentially an important additional component of efficacy of treatments, although not the primary focus of such trials, which tend to look at overall disability. In contrast, where a disease or disorder may be characterized by cognitive impairment, and here we think of things like Alzheimer's disease and other forms of dementia, or perhaps cognitive impairment associated with schizophrenia, cognitive assessments there may form the basis of primary or co-primary endpoints in clinical trials. A classic example of this, of course, is the use of ADAS-Cog in mild to moderate dementia. And here, ADAS-Cog has commonly been employed as a co-primary outcome measure at this stage of disease and used to support either global or functional outcomes as the other component of the co-primary, and confirm that treatments are having an effect on cognitive function in the patient population. And we can see here an example of ADAS-Cog improvement from a label for one of the cholinesterase inhibitors. We also know that in other trials in early stage disease where it's not possible to detect or measure global clinical impairment or functional impairment, that cognitive tests and test batteries, more specifically, are being developed and used as primary outcomes for such studies and used to track then disease progression over long periods in these early stage Alzheimer's disease patients. As a final consideration then, looking at surrogate and intermediate endpoints, one of the advantages there potentially is that we can have measures which can provide information about potential downstream clinical benefit, but which can be employed in studies which are perhaps over a shorter duration that may accelerate approval of a drug, perhaps with some post-approval commitments, but can help speed the drug development process where we have reliable predictors of later clinical benefit. In summary then, cognition assessments can be used to support decision-making in early phase development. They can form the basis of go-no-go criteria, particularly where we can look for consistency of evidence across multiple outcome measures and also set effect size benchmarks. In addition, in those early development phases, in some cases we can employ models of cognitive impairment that can also aid decision making and provide further evidence for potential cognitive efficacy. We know that we can use cognitive assessments in many different indications where they might provide supportive evidence of additional clinical benefit, and we looked at the example of multiple sclerosis. And also in some disorders of cognition, cognitive assessments may form the basis of primary or co-primary endpoints. And here we need to be really robust in terms of the extent to which we conduct validation and demonstrate that tests and outcome measures are valid, reliable, and sensitive to change for the context of use in which we use them. There may also be a possibility to develop cognitive assessments as intermediate or surrogate endpoints and as tools to increase the efficiency of drug development. So that concludes this brief overview looking at the application of cognitive assessments as efficacy outcome measures. We very much appreciate your attention, and we'd love to hear from you in case of any questions.